I'm sincerely thankful to the expert dynamic team at Cancer Research 2025 Magnus Group for sharing the screen and providing me a brilliant platform to start with the oral presentation for the day. Thank you for your expert assistance in uploading the presentation. Let's start with the talk. I'm Dr. Saumya Pandey, PhD Life Science, worked as Head Department of Clinical Research, Indira IVF Hospital, Udaipur, Lucknow, India. And the title for my talk is Autophagy as an immunomodulatory signalosome in toll-like receptors, Wnt-Frizzled mediated inflammatory gastrohepatic disease web in hepatocellular colorectal cholangio-carcinomas, translational research and public health impact in American cohorts of Texas, Nebraska, and New York states in United States of America. May I request the slide transition? Next slide, please. A quick cancer research snapshot. Gastrohepatic diseases, including colorectal cancer, are leading causes of morbidity and mortality in the United States, warranting cost-effective, evidence-based immunotherapy in at-risk susceptible population pools of varying genetic landscapes and lifestyles. I've published this in Hepatology 2017 and a paper from my side as the senior lead author in American Journal Physiology, Gastrointestinal Liver Physiology 2020. Further, gastrointestinal malignancies account for almost 1/4 of cancer-related deaths in the United States and approximately 30% of all cancer-related deaths worldwide, published by me in Asian Pacific Journal of Cancer Prevention 2015. Next slide, please. The complex autophagy machinery. This is from my publication in Asian Pacific Journal of Cancer Prevention in 2012. Cellular stress leads to a complex and intriguing biochemical cell signaling cascade with the interplay and complex crosstalk at the transmembrane and cytoplasmic level with PI3K, Beclin-1, BCL2, apoptosis, and the autophagy markers, autophagy-related 12, ATG5, ATG7, leading to the upregulation of the microtubule regulated light chain protein 3, which is the LC31 and LC32, two distinct isoforms, which are the markers for autophagy, a mode of cell death. And this has been implicated elegantly in colorectal carcinoma, and Beclin-1 happens to be the central player in the downstream mechanisms, including the transcriptional activation and nucleoplasmic shuttling, leading to the LC31 and LC32 altered flux in the physiological perturbed inflammatory hypoxic tumor core in the cancerous cell. And this has been implicated elegantly in colorectal carcinoma. So lots of dynamic collaborations going on globally in the ever-expanding field targeting the biochemical metabolic signaling pathway autophagy in conjunction with the enigmatic array of toll-like receptors biochemical signaling pathway, as well as the apoptosis pathway for tackling the ever-expanding burden of colorectal carcinoma worldwide. Next slide, please. A brief overview of autophagy in cancer immunotherapy. Autophagy, a process of cytoplasm and cellular organelle degradation in lysosomes, has been implicated in homeostasis, and under altered biological metabolic conditions such as cellular stress, the cell may undergo survival and/or cell death. So autophagy may in turn govern tumor metastasis and subsequent carcinogenesis in the physiologically starved, physiologically perturbed osmotic milieu of the cancerous cell, tilting the cell's fate towards cell death. And autophagy, one of the non-apoptotic cell death mechanisms, is characterized by engulfment of cytoplasm and organelles into double membrane-bound structures, the autophagosomes, which have the ATG2, ATG5, ATG7, ATG10, and the upregulation of LC31 and LC32, and subsequent delivery to and subsequent degradation into the lysosomes, leading to subsequent carcinogenesis. So autophagy in cancer immunotherapy has immense therapeutic potential for predictive and prognostic biomarker development in gastrointestinal malignancies, primarily colorectal carcinoma, in American cohorts of states of Texas, New York, Nebraska, as well as multi-ethnic cohorts of varying genetic landscapes and wild lifestyles. Next slide, please. Schematic depiction of autophagy. Transmembrane signaling, the engulfment at the membrane level, endosome formation, amphisome, autolysosome, the lysosomal degradation, phagophore formation, the initiation, elongation, involves the complex interplay of Beclin-1, which is the central player in the autophagy cell signaling metabolic pathway, and the conjugation of ATG12, ATG6, ATG16 complex with the vesicle closure, vesicle maturation, and subsequent degradation with the influx and outflux of LC31 and LC32, the autophagic flux getting tilted, leading to cell death in the inflammatory milieu in the cancerous cell. Next slide, please. We move on to the next slide, please. Immunobiology of toll-like receptors, emerging trends. This is my first scientific publication from Department of Biomedical Sciences, Creighton University School of Medicine, Omaha, Nebraska, United States of America, published in Immunology and Cell Biology on August 6, 2006. So a lot of thrust on the therapeutic manipulation of biochemical metabolic cell signaling pathways, primarily the autophagy and the enigmatic array of toll-like receptors, a family of evolutionary conserved pathogen recognition receptors playing pivotal roles as the primary sensors of invading pathogens, with TLRs identifying molecular motifs of infectious agents, the pathogen-associated molecular patterns, eliciting an effective defensive response against them. And mammalian TLRs derive their name from Drosophila tall protein, with which they share sequence similarity. And 10 TLRs have been known in humans. They work via the MyD88 signaling cascade and the TRIF-mediated pathway, leading to the NF-kappa B and interferon regulatory factor 3 upregulation and subsequent inflammatory cytokines and chemokines get upregulated in the physiologically perturbed inflammatory hypoxic tumor core in the cancerous tissue. So targeted manipulation of TLR signaling pathway in conjunction with the complex autophagic pathway involving Beclin-1, ATG, and the LC31 and 2 have immense therapeutic potential in the cancer research field for predictive and prognostic biomarker development in susceptible cohorts of varying genetic landscapes. Next slide, please. As I just discussed, toll-like receptors and their ligands, TLRs 1 to 10, an enigmatic array of receptors with TLR1 recognizing triacyl lipopolysaccharides, TLR2 the lipoprotein, PGNs, LTAs, sporins, and endotoxin lipopolysaccharide, lipoarabimonin, zymosin, trypanosomal phospholipids, and the heat shock proteins HSP60 and 70 defensins and MAM3 cis. TLR3 work via the TRIF-mediated pathway, and TLR4 follows the MyD88 as well as the TRIF-mediated pathway, leading to inflammation in the cancerous cells. And it is specific for endotoxin lipopolysaccharide along with HSPs, primarily HSP60, 70, and 90, hyaluronic acid, fibrinogen, heparin sulfate, and other TLRs, primarily TLR5, 6, 7, 8, and 9, have flagellants, diacyl lipopeptides, single-strand RNA, imiquimod, resiquimod, and bacterial viral DNA, and the CpG DNAs. So they recognize the pathogen-associated molecular patterns at the membrane level and subsequent downstream signaling pathway via different TLRs in conjunction with the pathogen-associated molecular pattern complex interplay, leading to inflammatory cytokines and chemokines upregulation and subsequent inflammation leading to tumorigenesis and metastasis, including carcinogenesis in the gastric hepatic system as well. So a lot of thrust on therapeutic manipulation of toll-like receptors and autophagy machinery in fighting the ever-expanding global challenge, the burden of the disproportionate share of morbidity and mortality, which can be attributed to gastrointestinal malignancies, primarily colorectal cancer. So here we are with precision-based, high-throughput therapeutic manipulation of TLRs and autophagy for predictive and prognostic biomarker development in colorectal carcinomas in multi-ethnic, genetically susceptible cohorts worldwide. Next slide, please. The complex TLR signaling pathway, ligand recognition, TLRs 1, 2, 3, 4, 5, 6, 7, 8, and 9, an enigmatic array of receptors working via the MyD88 and the TRIF-mediated pathway. And amongst the TLRs 1 to 10, 11, TLRs 3 specifically follow the TRIF-mediated pathway, leading to caspase recruitment and have been implicated in apoptosis and upregulation of type 1 interferons, while all other TLRs, basically TLRs 1, 2, 3, 4, 5, 6, 7, 8, TLR3 following the TRIF-mediated, follow the complex MyD88 signaling pathway with the conjunction of different sets of kinases such as TAB and TAC with JNK and AP1 phosphorylation, NF-kappa B upregulation, nucleocytoplasmic shuttling, transcriptional factor activation, leading to pro-inflammatory cytokines and chemokines in the hypoxic glucose-deprived tumor core in the carcinogenesis. So the enigmatic array of toll-like receptors and the complexity associated with toll-like receptor signaling pathway offer fascinating avenues for predictive and prognostic biomarker development and risk stratification in asymptomatic versus borderline versus symptomatic patients of gastrointestinal malignancies, primarily colorectal carcinomas, in individual cohorts of varying genetic landscapes and lifestyles. This has been published by me in Immunology and Cell Biology, 2006. Next slide, please. TLRs in immunotherapy. Lots of thrust on TLRs and autophagy machinery in cancer immunotherapy. The innate immune responses with cytokines and chemokines, defenses, and nitric oxide induction, endothelial responses, phagocytosis, eosinophil responses, adaptive immune responses, the upregulation of co-stimulatory molecules, primarily MHC class II on dendritic cells, T helper cells, designation, regulation, tolerance, cross-presentation of the CTLs, B-cell isotype switching and cross-presentation, detection of tissue injury constituting the sampling of commensals, recognition of host mRNA, heat shock proteins, as I already discussed in the previous slide with HSP 60, 70, and 90, matrix metalloproteinases, and tissue repair remodeling, primarily angiogenesis, involving the vascular endothelial growth factor, insulin-like growth factor receptor 1 and 2, actin polymerization, and apoptosis modulation, including the interplay of BCL2, endocrine responses, glucocorticoid production, and pathogenesis of sepsis, and pathogenesis even in allergic disease like asthma, along with autoimmune diseases, SLE, and diabetes. So TLRs have been targeted in a myriad human pathophysiology scenario, including diabetes, asthma, inflammatory diseases, including gastrointestinal malignancies, primarily colorectal carcinoma, and they provide valuable avenues, therapeutic avenues for chemoradiotherapy and vaccine strategies and biomarker development, such as the CpG-DNA anti-tumor and vaccine adjuvant TLR9, with lots of thrust on the interplay between toll-like receptors and autophagy and apoptosis in cancer immunotherapy for eventual precision-based medicine, precision oncology. Next slide, please. The Wnt beta-catenin signaling has also been implicated in colorectal carcinoma with secreted Frizzled-related proteins. Frizzled are a class of serpentine receptors, 7-pass receptors. They involve the Dickoff and the LRP5/6, leading to the complex formation of GSK, APCs, beta-catenin with the phosphorylation and ubiquitination leading to proteolysis and target gene manipulation, along with the Frizzled and Wnt conjunction, leading to the complex formation of axon disheveled glycogen synthase kinase 3, adenomatosis polyposis coli, APC, beta-catenin disintegration with the phosphorylation and dephosphorylation, nucleocytoplasmic shuttling, leading to the beta-catenin and transcriptional factor upregulation and downregulation, leading to targeted activation of the inflammatory cytokines and chemokines in the cancerous cell. So, Wnt beta-catenin, toll-like receptors, apoptosis, and autophagy complex signaling can be targeted together for precision oncology in colorectal carcinogenesis. Next slide, please. Objectives of my study included dissecting the complex cellular, molecular, and genetic regulatory biochemical signalosome, autophagy, toll-like receptors, Wnt-Frizzled, and inflammatory gastrohepatic disease web, primary hepatocellular colorectal cholangio-carcinomas is essential for diminishing the disproportionate share of morbidities and mortalities in susceptible at-risk American cohorts of Texas, Nebraska, and New York states in USA. Next slide. I aimed to investigate the potential immunomodulatory role of complex autophagy, toll-like receptors, Wnt-Frizzled signaling networks, crosstalks in pathobiology and prevention of gastrohepatic cancers, primarily hepatocellular, colorectal, cholangiocarcinomas, for eventual evidence-based design of promising patient-friendly, cost-effective predictive prognostic biomarkers and pharmacological scaffolds for novel drugs with minimal adverse effects for cancer immunotherapy. Next slide, please. Methods: cell viability. MTT proliferation assays were performed under basal and glucose-derived metabolic physiological conditions in toll-like receptor 4 agonist lipopolysaccharide endotoxin-treated cells in culture, primarily the HepG2, HD29, and the adenocarcinomatous SW480 CACO2 gastrohepatic colorectal carcinoma cells. Autophagy flux monitoring by assessing the relative ratios of LC32 versus LC31 isoforms in 0, 12, 24, 48 hours time course in glucose-derived triggered cells in absence and or presence of the TLR4 agonist LPS. Next. Cytotoxicity assays, semi-quantitative reverse transcriptase polymerase chain reaction, immunofluorescence, and DAPI-Hay staining were performed, followed by protein isolation by the radioimmunoprecipitation assay REPA method, protein estimation by Bradford's method, Western blotting with the primary antibodies from Cell Signaling Tech USA. Immunoblots were subjected to densitometric scans, and relative protein expression in my study with fold changes were determined with glyceraldehyde 3-phosphate dehydrogenase GAPDH and beta-actin as internal controls. Next slide. Results. The study yielded promising, convincing evidence-based results with HepG2, HD29, SW480 CACO2 gastrohepatic colorectal carcinoma cells, more than 80% viable under basal and glucose-derived metabolic physiological conditions in TLR4 agonist lipopolysaccharide endotoxin-stimulated sterile culture in vitro conditions. Autophagy flux was significant in gene-triggered starvation and hypoxic conditions with relatively higher expression levels of the autophagy marker LC32, which is 16 kilodaltons versus LC31, 14 kilodaltons in the strenuous time course experiments spanning 48 hours, 0 to 12 to 24 to 48 hours. Next slide. Wnt-124, 511 and Frizzled 125 yielded relatively low mRNA expression levels in HepG2 CACO2 cell lines. For the TLR4 agonist, LPS modulated the autophagic flux, which was significant in SW480 adenocarcinoma cells in the meticulous 48 hours time course with differential LC32 versus LC31 expression levels, the 16 kilodalton versus the 14 kilodalton autophagic flux titers. Interestingly, protein expression patterns of LC32 isoform was significantly higher than LC31 over 0, 12, 24, 48 hours in HepG2, HT29, SW480, and CACO2 cells with P less than 0.05. Next slide. Microtubule-associated protein light chain 3, the LC3 1 to 2 isoforms with the fold changes in HT29 and the SW480 adenocarcinoma colorectal cancer cell lines, the flux and the fold change with 16 kilodalton and 14 kilodalton, 16 kilodalton LC31 and 14 kilodalton LC32, GAPDH of 37 kilodalton was used as an internal control. So promising results with the microtubule-associated protein light chain 3, which is a known marker for autophagy. So indeed, under hypoxia, glucose deprivation, cellular stress, the physiologically perturbed stress cell with the altered osmolality, altered physiological milieu undergoes the tilt from survival to cell death with the upregulation of the microtubule-associated protein light chain 3 LC3 with the upregulation and downregulation of the LC3 1 and 2 isoforms. So autophagy flux leading to colorectal carcinogenesis, offering fascinating avenues for dynamic collaborations for future multicentric and multi-ethnic cohort studies in precision oncology for eventual evidence-based development of predictive and prognostic biomarkers, as well as therapeutic drugs for colorectal carcinoma management globally. Next slide, please. The ATG 5 and 7, 55 kilodalton, 78 kilodalton fold change, and 37 kilodalton was the GAPDH for the internal control for standardization. So we see that the HT29 and the adenocarcinoma SW480 cells, both the colorectal carcinoma cells, have the upregulation of the autophagic machinery, primarily the ATG5 of 55 kilodalton, with fold changes 1, 1, 0.9, 0.7 in HT29, and 1, 1.3, 1.2, and 1.0 in the SW480, significant fold changes and significant expression of ATG5 and 78 kilodalton ATG7 in HT29 and SW480, therapeutically sound, scientifically convincing data sets implicating the involvement of autophagic cell death in the physiologically stressed, perturbed, glucose-deprived cancerous cells, the HT29, as well as the SW480 adenocarcinoma cell. Next slide, please. Beclin-1, a central player in autophagy, 60 kilodalton, upregulation in HT29 and SW480 cells. We see significant levels of Beclin-1 with 1.0, 0.9, 0.71, and 1, 1.8, and 1.0 fold in HT29 and SW480. GAPDH, once again, 37 kilodalton for standardization under basal and glucose-deprived conditions. So Beclin-1 expression levels showing that, yes, autophagy, cell death is indeed occurring under the altered flux, under the altered physiologically perturbed, hypoxic, glucose-deprived, stressful cell in the gastrohepatic system, in the gastric cell, in HT29, and the adenocarcinoma SW480 cells, offering once again fascinating avenues for future design of pharmacological scaffolds in conjunction with predictive and prognostic biomarker design and development and novel drugs, logistic targets for carcinomas in multi-ethnic cohorts at risk of developing colorectal cancer with varying genetic landscapes and lifestyles. Next slide, please. SW480 colorectal carcinoma cell line glucose deprivation LC3 autophagy time course experiment. This was a meticulously designed, elegantly conducted time course experiment by me with autophagy marker LC3 detection by Western blot technique for glucose depletion GD as the autophagy trigger. The SW480 colorectal cancer cells were rinsed with glucose-free RPMI, which is the Roswell Park Memorial Institute RPMI 1640 media, followed by incubation in GD media glucose-free RPMI 1640 media with 10% heat-inactivated fetal bovine serum and 1% PS for 0 to 48 hours. Western blot analysis was performed to investigate the expression of LC31, a known hallmark of autophagy, with LC31 and LC32 isoforms, the A and B, 16 kilodalton and 14 kilodalton expression at 0 hours, 3 hours, 6 hours, 12 hours, 24 hours, and 48 hours, with GAPDH as the housekeeping internal standard. Next slide, please. Conclusions. My promising translational research study with emphasis on carcinogenesis strongly highlights the emerging immunotherapeutic potential of the complex autophagy toll-like receptors Wnt-Frizzled's biochemical metabolic signalosome in inflammatory hepatocellular colorectal cholangio-carcinomas for pharmacogenetics, genomics, metabolomics-based public health-oriented studies in ethnically disparate population subsets of the states of Texas, Nebraska, New York, USA. Next slide, please. Autophagy markers LC32 and LC31, the 16 to 14 kilodalton flux, appear promising therapeutic targets in cancer immunotherapy in the coming years for future development of patient-friendly, cost-effective predictive prognostic biomarkers, as well as pharmacological scaffolds and novel drugs for immunotherapeutically potent novel drugs with minimal adverse effects for tailor-made targeted cancer immunotherapy in susceptible at-risk cohorts of diverse ethnicities, dietary patterns, and lifestyles worldwide. Next slide, please. I have few impactful publications. Recently in gastrointestinal and hepatology research, an enriching snapshot published as senior lead author by me, Dr. Saumya Pandey, titled Toll-like receptor and Takeda G protein receptor 5 interplay in immunomodulation of inflammatory colorectal cancer and cholangio-carcinomas, cancer immunotherapy snapshot. Another paper on aldose reductase inhibitors in expert opinion on investigational drugs, focusing on toll-like receptors in carcinogenesis, along with the LPS endotoxin in the inflammatory milieu. Another paper in hepatology, which is from the American Association for Study of Liver Diseases, with a public health impact on non-invasive tests in advanced fibrosis due to non-alcoholic steatohepatitis, with critical insights from few clinical trials. And another recent paper in American Journal of Physiology, Gastrointestinal Liver Physiology, as the senior lead author on Takeda G protein receptor 5 signaling, mitigating parental nutrition-associated liver disease with public health impact. So lots of dynamic collaborations are warranted for leading experts and delegates today at our prestigious Cancer Research 2025 Singapore meeting for joining hands to design evidence-based, tailor-made, patient-friendly cancer immunotherapy regimen for eventual management, cost-effective management of cancer in susceptible cohorts of varying genetic landscapes. Next slide, please. I acknowledge National Institutes of Health, Bethesda, Maryland, USA for postdoctoral and doctoral biomedical translational research experiences at Schools of Medicine, University of Texas Medical Branch, Galveston, Texas, Creighton University School of Medicine, Omaha, Nebraska, and New York Presbyterian Weill Cornell Medical College, New York, USA. And I thank the audience today for a scientifically stimulating, dynamically collaborating discussion, and thank you to the prestigious Cancer Research 2025 Singapore Scientific Sessions team and the Magnus Group. And this is one of my first pictures with the first award, Dr. Saumya Pandey, that's me, first award at 4 years in hometown Lucknow at her school, La Martinia Girls College, Lucknow, India. So starting from a child and learning the intricacies of the complex, ever-expanding cancer research has indeed expanded my horizon. And I thank the Cancer Research 2025 Singapore prestigious Scientific Sessions Magnus Group team for providing me, Dr. Saumya Pandey, a scientifically enriching global platform for meaningful deliberation of ever-expanding thoughts for cost-effective management of cancer and cancer research on a global level. Thank you.